Hello everyone, and welcome to today's presentation. I'm excited to have you here as we explore the fascinating world of molecular cloning, genetic engineering, and recombinant DNA technology. On this first slide, we'll introduce the core concepts, starting with what cloning actually means, making identical copies of DNA. We'll look at how a specific gene can be isolated from a larger chromosome, attached to a carrier DNA to create recombinant DNA, and then transferred into a host organism like bacteria, virus, or yeast for replication. We'll also touch on how this technology allows us to produce proteins from genes that are normally expressed at very low levels, opening up many possibilities in research and medicine. Over the course of this presentation, we'll see how these methods enable us to sequence, characterize, and produce genes and proteins efficiently. So, let's dive in and explore these exciting topics together. Let's walk through the 5 key steps of cloning. First, we start by cutting the DNA we want to clone from the chromosome using a special enzyme called a restriction endonuclease. This enzyme is sequence-specific, meaning it recognizes and cuts at particular sites, which allows us to isolate the specific DNA segment of interest. Next, we select a cloning vector. This is usually a small, self-replicating DNA molecule, like a plasmid, that can multiply inside host cells. We cut this vector with the same restriction enzyme, which creates sticky ends or cohesive ends that are complementary to those on our target DNA. Then, we incubate the vector and the DNA fragment together. During this incubation, they anneal, meaning they stick together via those cohesive ends, and we join them permanently using an enzyme called DNA ligase. The result of this process is what we call recombinant DNA, which is a combination of DNA from different sources. The fourth step involves transferring this recombinant DNA into a suitable host, such as bacteria, viruses, or yeast. These organisms provide the necessary machinery for DNA replication, allowing us to produce many copies of our recombinant DNA. Finally, we need to identify which host cells actually contain the recombinant DNA. This step is crucial because it ensures we're working with the cells that carry our gene of interest, setting the stage for further analysis or production. Throughout these steps, it's important to understand that each one builds on the last, leading to effective cloning and gene manipulation, which is central to advances in biotechnology and medicine. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about cloning vectors, starting with circular plasmid DNAs, which are the most common type used in cloning. These are typically between 1 and 200 kilobases long and consist of DNA duplexes that contain essential genetic machinery. For example, they have a replication origin, which allows them to replicate independently within the host cell. This makes them very efficient tools for cloning. Plasmid vectors also carry specific genes that provide antibiotic resistance. This feature is crucial because it helps us select the bacterial colonies that actually contain the recombinant plasmids. After introducing the plasmid into bacteria, we can simply grow the bacteria on antibiotic-containing media, and only the ones with the plasmid will survive. Now, when we want to insert foreign DNA at a specific site within the plasmid, we often use a synthetic oligonucleotide that contains restriction sites for various restriction enzymes. This piece is ligated into the plasmid at a specific region called the polylinker, which is a sequence with multiple restriction sites. This design gives us flexibility to insert different DNA fragments precisely where we want. Beyond plasmids, there are other cloning vectors worth mentioning. Bacteriophage-based vectors are used for cloning larger DNA fragments, taking advantage of the phage's natural ability to infect bacteria. And then, for even larger genomes or complex studies, we have yeast artificial chromosomes, or YACs, which can carry very large DNA inserts and are useful for studying entire chromosomes or large gene clusters. All these tools, plasmids, phages, and YACs, are fundamental in molecular cloning because they expand our ability to manipulate and understand genetic material across different organisms. This slide doesn't contain enough information. That's why we're moving to the next slide. Now let's look at how two DNA fragments can be joined together, a crucial step in genetic engineering. Dale Kaiser and Paul Berg pioneered this by using an enzyme called terminal deoxynucleotidyl transferase, or TDT for short. This enzyme is pretty fascinating because it creates sticky or cohesive ends in DNA, which makes joining fragments much easier. TDT is a mammalian enzyme that adds nucleotides to the 3' end of DNA without needing a primer, meaning it can start from scratch. This allows us to generate specific overhangs on our DNA fragments. In practice, we take two DNA pieces and treat them separately with TDT in the presence of different nucleotides, one with DTTP to add poly-T tails, and the other with DATP to add poly-A tails. These complementary overhangs, the poly-T and poly-A, can then anneal or stick together due to base pairing. Once they're aligned, any gaps are filled in with DNA polymerase, and finally, the fragments are covalently joined using DNA ligase to create a stable, continuous DNA molecule. It's also important to note that TDT requires at least 3 unpaired nucleotides at the 3' end to work effectively. This can be achieved by using bacteriophage lambda exonuclease, which creates those free, unpaired nucleotides, setting the stage for TDT to add its tails. This process highlights how enzymes can be harnessed to manipulate DNA ends precisely, which is fundamental in cloning and genetic modification, connecting directly to our overall theme of DNA engineering techniques. This slide doesn't contain enough information. That's why we're moving to the next slide. Here we see a constructed E. coli plasmid called PBR322, which has been specifically designed for cloning in E. coli. This plasmid acts as a sort of vehicle, allowing us to insert foreign genes into bacteria efficiently. What's particularly important here is that PBR322 contains specific sites, like the PSDI restriction site, where we can cut and insert new DNA sequences. And as you'll see in the next slide, if we clone a foreign gene into this PSDI site, we can easily select for bacteria that have successfully taken up the gene. This setup is fundamental because it makes the process of cloning and selecting genetically modified bacteria much more straightforward. Overall, PBR322 is a powerful tool for genetic engineering, and understanding how it's constructed helps us grasp how we manipulate bacterial genomes for research and biotechnology. Now, let's talk about how we select the bacterial colony that contains our recombinant DNA. One of the key methods we use involves antibiotic resistance and sensitivity. Essentially, when we insert our gene of interest into bacteria, we often include a gene that confers resistance to a specific antibiotic. This allows us to identify which bacteria have successfully taken up the recombinant DNA. After growing the bacteria on a medium that contains that antibiotic, only the resistant colonies will survive and grow. Those resistant colonies are likely to contain our recombinant DNA. On the other hand, bacteria that haven't taken up the gene will be sensitive and won't grow in the presence of the antibiotic. This process is crucial because it helps us distinguish the bacteria that have the desired genetic modification from those that haven't. It's a straightforward, effective way to ensure we're working with the correct cells and moving forward with the right bacterial clones. So, in the bigger picture, this selection process is a vital step in genetic engineering, helping us efficiently identify and isolate the bacteria that carry our recombinant DNA, and ultimately, enabling us to study or utilize those genetically modified organisms. Let's talk about the bacteriophage cloning vector. This virus is incredibly efficient when it comes to delivering its DNA into a host bacterium. Specifically, it can carry about 48 kilobases of DNA, which is quite substantial. Now, what's interesting is that roughly 1/3 of its own DNA is non-essential. That means we can replace this non-essential part with foreign DNA, allowing us to insert our genes of interest without disrupting the virus's ability to infect. Once we've created this recombinant DNA, The next step is packaging it into a phage particle. To do this, we add our recombinant DNA to a bacterial extract that contains the necessary proteins for packaging. These proteins help assemble the DNA into new phage particles, ready to infect new bacteria and deliver the foreign DNA efficiently. This process highlights the power of bacteriophages as vectors. They're highly effective tools for gene cloning and manipulation, fitting seamlessly into our overall goal of harnessing viruses for genetic engineering. Now, let's talk about how we construct a complementary DNA, or cDNA, library. Essentially, cDNAs are DNA molecules that have sequences complementary to mRNA. This means that the cDNA represents the genes that are actively being expressed at the mRNA level in the cell. To start, we first isolate the mRNA, and we do this using an oligo-DT column. The reason for this is that the poly-A tail of mRNA binds specifically to the oligo-DT primer, allowing us to separate mRNA from other RNA types. Once isolated, the mRNA is annealed with an oligo-DT primer, setting the stage for reverse transcription. Next, we generate the cDNA using an enzyme called reverse transcriptase, along with dNTPs, which are the building blocks of DNA. Reverse transcriptase synthesizes a complementary DNA strand based on the mRNA template. After the first strand is made, the original mRNA is typically degraded by alkaline hydrolysis, leaving us with a single-stranded cDNA. We then use DNA polymerase I and additional dNTPs to synthesize the second DNA strand, resulting in a double-stranded cDNA molecule. The final step involves cloning these cDNAs into appropriate vectors, such as plasmids or phages, which can then be introduced into host cells through transfection. This process allows us to create a cDNA library, a collection of cloned cDNAs representing the actively expressed genes in a cell or tissue. By understanding this process, we see how cDNA libraries are powerful tools for studying gene expression, isolating specific genes, and even producing recombinant proteins. This step is fundamental in molecular biology research and has broad applications in biotechnology and medicine. Let's walk through how PCR amplifies a specific DNA segment. First, we start by heating the DNA to separate the two strands. This step makes the DNA single-stranded and ready for copying. Once the strands are separated, we cool the mixture down. At this stage, we add synthetic oligonucleotide primers that are designed to flank the region we want to amplify. These primers bind specifically to their complementary sequences on each strand. Next, we introduce a special enzyme called thermostable DNA polymerase, often Taq polymerase. This enzyme is crucial because it synthesizes a new DNA strand by adding nucleotides in the 5 to 3 direction, starting from the primers. Then we repeat this entire cycle, heating to denature, cooling to allow primer binding, and extending with DNA polymerase, about 30 to 40 times. Each cycle doubles the amount of target DNA, resulting in thousands, even millions, of copies of the original segment. This process is fundamental in molecular biology, enabling us to generate enough DNA for analysis or experimentation, and it exemplifies how precise, automated steps can exponentially amplify genetic material. Now, let's talk about how we screen our bacterial plasmid library using colony hybridization. First, we plate the bacteria that contain our cDNA library onto agar plates with the appropriate media, allowing colonies to grow. Then, we press a nitrocellulose, or NC, paper onto these colonies. This step transfers some bacteria onto the paper, capturing the genetic material. Next, we treat the NC paper with an alkali solution to lyse the bacteria, which exposes the cDNAs. The DNA from the colonies binds to the NC paper, and this is crucial because it preserves the genetic information for detection. To find the specific gene we're interested in, we use a radioactive DNA probe that's complementary to our target gene. This probe hybridizes, or binds, specifically to the matching cDNA on the NC paper. Finally, after exposing the hybridized NC paper to X-ray film, the areas with the desired gene show up as dark spots. This allows us to identify the colonies harboring the specific cDNA we're after. This process is a powerful way to screen large libraries efficiently and connect the dots between the bacterial colonies and the genes we want to study. Now, let's delve into DNA microarray analysis for gene expression. DNA chips, which contain spots with DNA from known genes, are commercially available, but you also have the option to create one with your desired DNA sequences. The process starts by isolating mRNA from both control and diseased tissues. Next, we convert this mRNA into complementary DNA, or cDNA, using fluorescent nucleotides, different colors for each sample, so we can distinguish them. These fluorescently labeled cDNAs are then hybridized with the DNA microarray chip. When hybridization occurs, the fluorescent spots on the chip indicate the expression levels of the corresponding genes. The intensity and color of these spots give us valuable information about how gene expression differs between the control and diseased states, helping us understand underlying biological processes. This technique is a powerful tool for comparing gene activity across different conditions, tying directly into our broader discussion on gene regulation and disease mechanisms. Let's talk about site-directed mutagenesis, a really powerful technique in genetic engineering. Michael Smith from Canada was awarded the Nobel Prize for pioneering work in this area, which highlights its significance. Essentially, this method allows us to introduce specific mutations into a DNA sequence, so we can make precise changes at a targeted spot in the gene. One common way to do this is by synthesizing a mutated version of the DNA and then inserting it into the original gene. This is typically done using restriction enzymes, which cut DNA at specific sites, and ligase, which helps to glue the pieces together. But the most popular procedure involves working with single-stranded DNA. Here, how it works. We create an oligonucleotide, basically a short piece of single-stranded DNA, that contains the desired mutation, a single nucleotide change. We then use this oligonucleotide as a primer in a DNA polymerase reaction to produce a mutant copy of the gene. This approach is especially valuable because it allows us to make very precise, targeted changes, which is crucial for studying gene function or developing new therapies. It connects directly to our overall theme of genetic manipulation, showing how we can engineer DNA with incredible specificity. Now, let's look at how we create recombinant plants using a fascinating technique involving a plant parasite called Agrobacterium, along with a 2-plasmid strategy. Essentially, Agrobacterium is a natural genetic engineer. It has this unique ability to transfer DNA into plant cells. By harnessing this natural process, we can introduce desired genes into the plant genome. The 2-plasmid strategy involves using 2 separate DNA molecules, one carrying the gene of interest and the other containing genes necessary for the transfer process. When combined, these plasmids enable precise insertion of the new genetic material into the plant, making this method highly effective for plant genetic engineering. This approach is a cornerstone in developing genetically modified crops, connecting directly to our overall theme of advancing agricultural biotechnology through innovative genetic tools. Here we see a fascinating example of genetic engineering in action. This is a tobacco plant that has been modified to express the gene for firefly luciferase. What this means is that the plant now produces the enzyme responsible for bioluminescence in fireflies. When we water the plant with luciferin, which is the substrate for luciferase, the plant actually glows. This highlights how we can introduce specific genes into plants and observe their activity through such visible traits. It ties into the broader theme of how genetic modification allows us to understand and manipulate biological processes in innovative ways. Now, here we see an exciting development in agricultural biotechnology. Tomato plants that have been engineered to be resistant to some insect larvae. What this means is that scientists are modifying the plants at a genetic level to produce certain traits that can deter or even defend against pest attacks. Essentially, these plants are better equipped to survive and thrive without the heavy reliance on chemical pesticides. This approach not only helps improve crop yields, but also promotes more sustainable farming practices by reducing chemical use, which is better for the environment and farm workers. It's a great example of how genetic engineering aligns with our broader goal of creating more resilient and eco-friendly agricultural systems. By integrating pest resistance directly into the plant, we're moving toward a future where crops are more sustainable and productive, addressing some of the key challenges faced in modern agriculture. This slide doesn't contain enough information. That's why we're moving to the next slide. Now, while cloning offers some exciting possibilities, it's important to also consider its negative features. For example, one major concern is the genetic destruction of plant germplasm, which means we could be losing valuable genetic diversity that's crucial for resilience and adaptation. Ecological imbalance is another serious issue, since introducing cloned organisms into ecosystems might disrupt existing natural relationships and processes. Then there's the production of monsters. This refers to the creation of genetically modified organisms, or hybrids, that could have unforeseen and potentially dangerous consequences. We also face the risk of producing highly toxic chemicals and microbes that can be lethal. Such dangerous substances might be developed intentionally or accidentally, and their misuse could lead to devastating outcomes. This ties into the darker aspect of microbiological warfare, using lethal microbes as biological weapons to harm humans, animals, and plants. Overall, these negative features highlight the importance of careful ethical considerations and regulation when it comes to cloning technology, especially as it relates to environmental and health safety.